Hi everyone, today let's continue the discussion about the trafficking of the leukocytes, right? When you talk about leukocytes, you know, it's monocytes, neutrophils, lymphocytes, and so forth. So if you close your eyes and try to imagine these cells, they need to travel through our body in order to fight the pathogen or to repair an injured tissue and so forth. So these cells cannot stay all the time in the same place like the other cells of our tissues and organs. So the immune system as a whole, it's a dynamic system. The immune cells are constantly traveling. And if you imagine all this traveling needs to be extremely coordinated in order to exert its proper functions. So how does the body communicate with the immune system to say, hey, I want this immune cell here now to help me to fight this infection, and then afterwards you can go away? How does that happen? It happens through the trafficking properties of the leukocytes, the immune cells or leukocytes. This is actually a very beautiful process, and we call that the rolling of the leukocytes. So in order to happen the proper trafficking of the leukocytes so that they can arrive in a particular tissue of need, we need to bear in mind two main steps that need to be accomplished. First, the attraction of these leukocytes, and second, the adhesion. They are not only attracted there, but they stay there. Then the tissue or the organ communicates with the surrounding blood vessels where the immune cells are traveling, with the surrounding blood vessels, and then they say, hey, you need to attract the leukocytes that are arriving here so they stay here. So the organs tell that to the blood vessels. And the blood vessels are constituted by endothelial cells, right? That's the name of the cells that constitute the pipes of the blood vessels, endothelial cells. So when there's a need, when there's an infection, when there's an injury, the endothelial cells, in response to the needs of the tissue, will start producing a chemical, a substance that will attract the coming leukocytes to stay there. Is an attraction. That's the first signal. And the leukocytes, they have receptors for this chemokine. So when they receive this chemokine, it's produced there, when they arrive in that area that's full of these chemokines, it will attach, it will bind to the receptors of this chemokine expressed in the leukocytes, in the immune cells. And then the immune cells will say, hey, I need to stay here. There's something happening. I'm feeling that I need to do something here. They have in the surface of their cells, in the cell membrane, adhesion proteins, but when they are traveling, these adhesion proteins are closed. They have low affinity to their receptors. So when the chemokine binds its receptor in the immune cell, it starts a signaling cascade process that will ultimately educate the leukocyte to tell these adhesion molecules in the surface that now you need to relax, extend, and acquire high affinity. So initially, the adhesion molecule was bended and with low affinity, now is extended with high affinity. And then these integrins will bind to the receptors that are expressed there in these endothelial cells, and they stay there. They stop traveling. The heart will keep pumping, the other cells will keep traveling, but the ones that found the chemokine and they express these integrins with high affinity, they start binding the tissue to infiltrate the tissue. It's a beautiful process. And that happens like programmatically while the cells are traveling, they are going through the pipes, it's full of chemokines here, they start rolling, the chemokines bind to the receptors of chemokines in the immune cell, and then the immune cells start expressing these high affinity integrins, and they start attaching here while they are rolling through from the endothelial cells, and they stop. And then they penetrate the tissue through the endothelial cells. The endothelial cells open and the immune cells infiltrate the tissues so they can exert their effector functions, right? And of course, there are names for these integrins, for the receptors of the integrins, there are names for the chemokines, there are different names for the receptors of the chemokines. So the low affinity integrin, once it becomes with high affinity, it binds, for example, to icon 1. It's a ligand expressed in the endothelial cells that will bind to the high affinity integrin so that the leukocyte will stay there. So the chemokines, they activate the adhesion of the leukocytes and then the leukocytes start migrating towards a gradient of these chemokines, right? It's actually a process that is called chemotaxis, or this chemoattraction is a chemotaxis of these leukocytes, and they not only attach to the tissue, but they can also migrate in the direction where you have higher concentration of these chemokines being secreted. This is how you educate the migration, the translocation of the immune cells from the bloodstream and then inside of the tissues. Because inside of the tissues, they also need to know where to go. So the chemoattraction will tell them where to go, and they go towards the gradient, the concentration gradient of these chemokines produced inside of the tissue as well. So what are the chemokines? The name chemokine is a combination of chemotact with cytokine. Cytokine is a protein produced by the immune system to exert its functions like a hormone, right? The chemokines, they are a large family of structurally and homologous cytokines that are produced to induce the traffic of the leukocytes from the blood to the tissues. "-ine is the suffix that determines a protein, so cytokine is a protein. But chemokines are not complex proteins. A complex protein would be, for example, insulin. If you look to the structure of an insulin, it has several different turns and folding. It doesn't look like something symmetric. It has actually several different substructures there. It's a very complex molecule. But if you think about a cytokine or a chemokine, this chemotact combined with a cytokine, chemokine, is actually a very simple protein. It's composed by fragments of proteins. It's composed by a polypeptide. It's usually between 8 to 10 kilodaltons. Kilodaltons is like the metric that we use to rank the size of a protein. So these chemokines, they are not very complex. They have like a few turns. If you think about the tertiary structure of these proteins, they're like polypeptides with a few turns, and these turns are usually bonds of cysteine, which is a very reactive amino acid that then they attract to each other. They create these very strong interactions, these cysteine bridges, and they will make these turns, one turn here and there. They create this small complex, let's say, peptide that has a particular function. It's kind of like, it looks like a hormone, right? So it's not a very complex protein, but it has a particular function, a particular tertiary structure to bind that small receptor to activate a cascade of events in the immune cell, right? Amino acids, they compose the protein. The small fragment of a protein itself is the amino acid. So the combination of different amino acids will create a protein. But when you have like a cysteine, it's one of the amino acids. It starts with a C. So a cysteine can interact with another amino acid in the same protein, creating bridges. And this is a very strong interaction. So when you have a cysteine interacting with another cysteine, it creates this interacting bridge, and the protein folds when that happens, and that determines, for example, the tertiary structure of the chemokine. So depending on the arrangement on the number of cysteine connections inside of the chemokine, it will determine the type of family that this chemokine belongs to. We have in total 47 chemokines in our body, in the human body. We have 47 chemokines produced by our cells, divided into 4 families. And these 4 families are divided in 2 major families. The first one groups the type of chemokines where the cysteines that are connected to each other, they are adjacent. So when you look into the amino acid sequence of this protein, these 2 cysteines are connected, they are adjacent to each other. So this is one type of chemokine. It's named like CCL something. And the other major family of chemokines include those chemokines where the cysteines are separated by one amino acid. When you look into the amino acid sequence, there's the cysteine here and then one amino acid and another cysteine. So it could be random, any amino acid, so it's X amino acid in the middle of these 2 cysteines. It will also fold and create a tertiary structure that will bind to one particular receptor, but this is the main signature of this type of chemokines. So the preserved and conserved cysteines interacting to one another, they are interacting, but there's an amino acid in the middle. So they are named or classified as CXC, CXCL something. L comes from ligand. So CXCL1, CXCL12, and so forth, right? So the first one is a major family. It's characterized of chemokines CC something, and the other one CXC something. So for example, the chemokine CCL2, the original name of this protein is MCP1, but it belongs to the family of chemokines, so it has a label of a chemokine, is CCL2. L comes from ligand. It will usually bind to a receptor in the leukocyte, a receptor that is specific to this chemokine, the CCR2, the receptor for CCL2. So CCL2 is mostly responsible for a mixed recruitment of all leukocytes. And another example is the CXCL1. It is responsible for the recruitment of neutrophils when there is a site of infection. So I hope that you liked this lecture. If you have any questions, don't forget to write them in the comments below. I'm more than happy to come here and answer for you. And also please help me to disseminate this channel and to share them with your friends, colleagues. Let's teach immunology to everyone. I'll see you in the next video. Bye-bye.